The Krebs cycle is an important part of cellular respiration. It has not only one but three names: the Krebs cycle, after Hans Krebs, who discovered it; the citric acid cycle, named for citrate, the first molecule forming during the reaction; or citric acid, which is the protonated form of citrate. It can also be called tricarboxylic acid cycle, or TCA for short, named for the three carboxyl groups on the first two intermediates in the cycle. The citric acid cycle takes place within the inner membrane of the mitochondria. Since the Krebs cycle is an aerobic process, it takes place only within eukaryotic cells. For the Krebs cycle to happen, glycolysis needs to occur. Glycolysis is when a 6-carbon glucose is broken down into two 3-carbon pyruvate molecules. Following glycolysis, pyruvate needs to be transformed into acetyl-CoA. Each pyruvate molecule loses one carbon atom and releases carbon dioxide. During this breakdown of pyruvate into acetyl-CoA, electrons are transferred to NAD+ to produce NADH, which will be used by the cell to later produce ATP. In the first step of the citric acid cycle, acetyl-CoA joins with a 4-carbon molecule, oxaloacetate, and releases a CoA group and forms a 6-carbon molecule called citrate. In the second step, citrate is converted into its isomer, isocitrate. This involves the removal and addition of a water molecule. After isocitrate is formed, it's oxidized, and it then releases a molecule of CO2 and leaves behind a 5-carbon molecule called alpha-ketoglutarate. During this step, NAD+ is reduced to form NADH. The enzyme catalyzing this step is isocitrate dehydrogenase. The fourth step involves alpha-ketoglutarate dehydrogenase in a very similar fashion to the previous step. Alpha-ketoglutarate is oxidized, reducing NAD+ to NADH, again releasing a molecule of carbon dioxide. The remaining 4-carbon molecule gains coenzyme A, forming an unstable compound known as succinyl-CoA. CoA of succinyl-CoA will be replaced by a phosphate group, which will be transferred to ADP to form ATP. In certain cells, GDP will be used instead of ADP, which will form GTP. CoA of succinyl-CoA being replaced by a phosphate group results in a 4-carbon molecule called succinate. Succinate being oxidized forms another 4-carbon molecule called fumarate. This process leads to 2 hydrogen atoms and their electrons being transferred to FAD, producing FADH2, the 4-carbon molecule known as fumarate, yet again converting it to a 4-carbon molecule called malate. And the last step, oxaloacetate, which is the starting compound, is regenerated by oxidation of malate. Another molecule of NAD+ is reduced to NADH in this process.